So recording is also started. So sorry for the delay in starting today's class. Thanks, Murli, for giving message. So in today's class, we will finish up the topic coordination in cellular fluid processes. So in the last class, we saw how the ECM, cellular matrix, it helps in coordinating the cellular fluid processes. Like the components in the ECM, they control the fluid processes, and the ECM in turn is modulated, like changed the component composition, and like it is over time secreted, new components secreted, and uh, other compounds are degraded. So this maintenance of the ECM is done by the cells. There's a cyclic kind of a process going on. And we also saw what kind of integrant-based junctions are there that the cell is forming with the ACM components, and what are their roles, etc. Now we will see what happens if there is malfunctioning going on. Okay, so as with soluble signals, the signaling cues they provided by the ECM, they regulate cellular behavior, all right, and as well as they control the tissue function and the homeostasis. Just like soluble signal does, similarly ECM can also do this, like control cellular function, the tissue function, and homeostasis. But if there is some mutation in the genes that encode for various kind of ECM proteins like fibronectin, leukocyte, etc., or the ECM remodeling proteins like MMPs, matrix metalloproteases proteins, or the ECM receptors. Okay, the cell binding domains that is there in the ECM, and in the cell there are receptors like integrin receptors. So if in these three things there is some mutation, okay, yeah, and the product, the protein product that is made is a mutant one, so it can cause some diseases, right? So people have observed mutations, and they have seen mutations in, say, the class of matrix metalloproteases. Like people have found uh, the how the mutations in this family of protein is linked to various kind of diseases. So also there is particular interest in MMPs as they are also involved with the development of the cancer. So various kind of cancers are related to or have developed because there is some mutation has come up in some kind of a MMP protein. And the dynamic degradation of synthesis of chondrocytes takes place, right? We have seen one case study where the chondrocytes they secrete the same component in response to the normal forces that is being applied. So there is a dynamic synthesis as well as degradation is going on in response to the forces. So there will be always a synthesis of the enzymes that degrades the ACM components, as well as there are biochemical pathways that synthesizes the ACM proteins. And there is a fine balance that is needed in between these two processes. And if this balance is lost, synthesis of new ACM components and the degradation of the old one, if this balance is lost, then some pathological conditions can arise. So here, this is what happens during development. Means so one this is basically a swing, okay, or you can say a balance where on one side there are MMPs, okay, and there's and the other side there is your ECM protein. So during development, like during embryonic development, the activity of these MMPs is very high, all right, because and they are very functional. That is what I'm trying to say. Because the cells are continuously changing their orientation and new tissues are being formed. So obviously, synthesis and degradation process is happening at a dynamic rate. But when the tissues have achieved the homeostasis condition, like in normal adult tissues, then there is a fine balance. The degradation by the MMPs and the synthesis of ACM proteins, they should be in a balance, right? And when the tissue is damaged and the repair process needs to take place, it's a wound healing process, there also during the remodeling phase, if you remember, the ECM components, they are changing their orientation, and the composition of various type of ECM components is changing. So then also the activity of MMPs is increasing. Now, suppose during normal condition or normal physiology, if the balance is lost, okay, MMP is much more active than the degrading completely the ECM components, whereas the other side, the synthesis of the new ECM components is not happening at the fine balance. Means when this balance is lost, then a pathological condition like cancer can develop. So this can happen due to mutation in MMP protein, which can lead to overexpression of that MMP protein, and thus uh, the composition of the ECM of the tissue is changing rapidly. Right. So the balance between these two things is very important for maintaining a homeostatic condition of a tissue. Now let's take an example, okay, to understand how ECM molecules or ECM components, they are actually controlling cellular function like through some proteins, and how in turn these protein is changing the ECM composition. So if anyone, if you have like read cancer biology or similar course like that, you must have heard the proteins RB and P53. These are basically tumor suppressor genes, and these are very important proteins. And P53 is actually an oncogene, and its role, it is very important protein for cell cycle as well. Means it's one of the checkpoint proteins as well, P53 and RB. So these two proteins, they regulate, okay, they regulate and as well as interact with the ECM component, and it happens in a feedback loop kind of a mechanism. What happens is the phosphorylation of RB protein and the transcriptional activity, translocation, and degradation of P53 protein. These two functions, mean these two activities, they are regulated by the interaction between integrin cell antigens that are present in the cell membrane and the ECM components. Means interaction between integrin and ECM component to regulate the function of RB and P53. And in turn, RB regulate the expression of the ECM remodeling MMPs, okay, and P53 regulate the state of the ECM through transcriptional control of ECM components and mediate cell ECM signaling as well as ECM remodeling. Okay. So RB is controlling synthesis of the MMPs, and P53 is basically controlling the synthesis of new ECM components, okay, as well as regulating cell ECM interactions. So this is a feedback kind of a thing going on, feedback loop. So here, this is the cell membrane. So on the cell membrane, there are integrins present. So when cell and ECM interaction takes place, that in turn is regulating the activation. RB gets activated when a phosphate group is attached to it, and it's get deactivated when phosphate is removed. So it is controlling the activation of RB. Once RB protein is activated, it is going to control the synthesis of the MMPs, that is matrix metalloproteinases. That in turn will remodel or help in remodeling of the ECM, means degrade ECM components, etc. Whereas again, T cell ECM interaction is also controlling the activity of P53. How? It is controlling the synthesis, okay, of these many types of ECM proteins. So these are ECM proteins, they're synthesized by the cell only. The synthesis pathway at the transcription level, P53 is controlling, okay, and which in turn is controlling the ECM composition, overall ECM composition. And it is also, this P53 is also playing an important in controlling the activity of the MMPs. So this is a feedback mechanism between the two tumor suppressor genes, P53 and RB, and the ECM component. So if suppose P53 gets mutated, okay, and is dysfunctional, then you can understand it can severely affect the composition of the ECM and overall function of the cells and the tissue overall. Okay, and if suppose the active gene for the MMP is mutated somehow, then also the cell ECM interaction will get hampered, and the cell will not get the normal physiological signals, and thus its activity will start malfunctioning, and it even can become cancerous. So when a balance in this kind of or if this kind of feedback loops get disrupted, then malfunctioning in ECM signal can result. Now, another way through which cell are in between the cells, the coordination of the cellular processes happens is through direct cell-cell contact. So cell is in contact with the ECM in a tissue, as well as cell is in contact with other neighboring cells, right? Tissue is composed of cells and tissues, and cells can communicate with other cells by secreting some soluble signals, but it can also directly communicate with the cells which is directly adjacent to it. So, through soluble signaling, it can communicate with a larger population of cells, let's say 10 cells, means at whatever distance the signaling molecules can diffuse to that distance, how many cells are there? There is 100 cells, 10 cells, or even thousands of cells. At a time, it can communicate with that number of cells. But direct direct cell contact, at a time, one cell can communicate hardly with two or three cells. Or if you consider it as a square, maybe four edges. At four edges, how many cells are there? It can communicate with them. So the reach of the cell is decreased when you are thinking about direct cell-cell contact. But this kind of communication is very specific. Okay, it's a very specific mode of communication, and because obviously uh, the cell knows to which cell it is discussing whatever information it is passing, right? So it becomes highly specific. The target is highly specific. Only that adjacent cell, the neighboring cell, will receive the signal. And whereas the soluble signaling and recent signaling is non-specific. Okay? So here the target cell is not decided by the signaling cell. It can it depends on the soluble signals, like how far it can diffuse, or it depends on the composition of the ECM. And ECM is present everywhere in the tissue. So which ECM component will bind or signal which cell, it is highly out of control of the source cell, which has secreted the ECM component. So the target cell, it has no direct knowledge of the uh, signaling cell. Also, the signaling cell has no direct knowledge of what is going to be the target cell. So the direct cell-cell signaling, it can happen in two ways. One way is that there is a mechanical contact between two cells, and another way is that there is a junction that is present between two cells through which the molecules can directly pass from one cell to the other. Okay, so one is like tight mechanical contact where there is no passage for passing through of any molecules. Another contact is the passages are there, okay, of fixed diameters that only allows molecules of fixed diameters. So very small molecules can only transport from one cell to another. One second, I have a call that I need to receive. Just give me one second. I'm sorry for the interruption. So, as I was saying, that is, there are two types of direct cell-cell contact that is present. One is mechanical contact, another is a junction, which is having a hollow passage through which the molecules can pass. So, as I was saying, the two classes of connection that is there between two cells. So this, so if we again closely look into what kind of cell junctions are there, we can categorize it into four junctions. So one, two, and three: tight junction, adherent junction, and desmosome. These three junctions allow mechanical contact, whereas the fourth one, the gap junction, here hollow passage is there through which the molecules can pass. So if we look diagram of how the junctions look like, so this is these two cells. Okay, these are two cells. The first one is a tight junction, where it's like a barrier. Okay, like a very strong glue, and it doesn't allow any tissue leakage or anything like that, or leakage of any cytosolic components. So this is a tight junction. And then there is adherence junction. Here, the cytoskeleton actin filaments, they are linked. Means actin filaments of this cell and this cell, they are linked with each other through adherence junction. So again, a mechanical contact between the cell is formed. In desmosome, here the keratin intermediate filaments, those are linked. Okay, this cell for this cell and this cell through the desmosomes. Whereas in gap junction, here you can see a passage is there through which few molecules can can get easily transported from this cell to this cell. And other type of linkages that you know, the cell can bind with the ECM through the hemidesmosomes or the focal contacts, the fibular contacts that allow connection between the actin and the keratin filaments with the ECM. So these are cell matrix junctions that you're already familiar with, and these are cell-cell junctions that we're studying now. Now, let's briefly describe all the four types of cell junctions. So, the tight junctions, they are an impenetrable barrier, okay, that effectively fuse the adjacent cell membranes together. So, as I said, they prevent any leakage, okay, a cell tissue leakage or the cytosolic protein leakage out. That is the role of tight junctions. So they are like a seal that gives a tissue a barrier function. So which kind of tissue you think needs a barrier function? So one example I can think of is a skin. So skin, the role of skin is to protect us from external, protect our internal organs basically. Okay, so from any kind of chemicals or even UV, etc. So it's a barrier tissue for us. So the epithelial, topmost layer, the epidermis. So that cells in that layer is going to have the tight junctions. That is going to overall give the barrier function to the tissue. So you can definitely think of other epithelial layers like in our gastrointestinal tract, okay, like in our stomach. So a very nice epithelium layer should be present, smooth epithelial layer should be present that will prevent the leakage of acids that gets formed in our stomach. So these, for fulfilling these kind of functions, these tight junctions at the cellular level are doing the job. Then the adherent junctions. So these are also called belt desmosomes. And what they do, they join the actin filaments of one cell to a similar bundle of actin filaments in the neighboring cell. So they are like a backup, okay, and backup to the tight junctions. and they provide mechanical strength to the tissue. Okay? Also regulate tissue permeability. Permeability means it regulates passage of any molecules like water, etc. Cells, if kept in different kind of solutions, hypertonic, hypotonic solutions, then water molecules can easily diffuse out or diffuse in. So this is permeability. Our cell membrane is semi-permeable. So this level of permeability, I gave an example or gave example of water molecules. Other type of molecules can also enter. So this regulation is done by the adhering junction. Then another type of cell junction is desmosome junction. These are also known by spot wheels. So the role of this junction is again to provide mechanical strength, means mechanically connect the cells of the tissue. And here the keratin intermediate filaments of one cell is connected with the other cell. And these filaments, intermediate filaments, they are connected to the ECM via hemidesmosomes. So the role of cytoskeleton, you can understand now to give a framework to the cell, okay, and connected to other structures like the cell and the ECM, so that the placement of the cell is maintained, as well as it is given the cell overall is having some mechanical strength. It cannot get deformed very easily if it gets binded with the ECM and other cell. So the role of actin as well as the keratin intermediate filaments. So these cytoskeletal uh, role of the cytoskeletal structures you might be realizing now. You might have studied long back in your plus 2 level, but why it was important now you might be realizing. Now, another type of cell junction is the gap junction. Now, these are the hollow junctions, okay? And they allow passage of very small molecules like, say, water, okay? Water soluble ions, so water soluble ions like, say, sodium, potassium, okay, calcium ions. So these ions only can pass through, and the limit of the size is given by less than 1 kilodalton, equal to a less than 1 kilodalton. So very small molecules can only pass through these gap junctions. So these are four types of cell junctions. Three of them are for providing mechanical strength to the cells and the tissue, and the gap junction is for passage of small molecules which are of very small size, size less than 1 kilodalton or equal to 1 kilodalton. So, any queries so far? Is anyone there in the meeting?
No, ma'am, no doubt, ma'am.
No doubt. Okay. So, anyone else having any doubt? Everyone physically present or virtually present? Yeah.
Can you repeat the function of the desmosomes?
Okay. So, Aditya, thank you for this question. So, desmosomes is a cell-cell junction that is giving strength, okay, mechanical strength to the cells. Their role is to connect the intermediate filaments. So, what are intermediate filaments? Just cytoskeletal elements. So, they connect the intermediate filaments of one cell with the another. That's it. Its role is to just provide connection between the intermediate filaments of the neighboring cell. And these intermediate filaments, if you recall, they are also linked with the ECM through another integrin-based junction, that is through hemidesmosomes. So in this diagram, you can see these are tight junctions, these are adhering junctions. Here the actin filaments are linked of one cell to the another cell, and desmosome is connecting the intermediate filaments. Okay. Is
Okay,
your
thank you, ma'am.
doubt clear? Okay.
It's fine.
Any other queries? All right. So let's move on. The molecular components in cell junctions. So let's see that. Now both desmosome and adherent junctions they utilize the membrane-bound receptors of cadherin family. So let's talk about the family of proteins that are involved in forming these kind of cell-cell junctions. So we will see that. So here, desmosomes and the adherent junctions they utilize the family of protein that is called cadherins. So in this figure, you can see. So this is the membrane of one cell, this is the membrane of other cell. So these are the cadherin family of protein that are involved in forming adherent junction and desmosome junction. So cadherin family of protein is connected with either actin filaments or intermediate filaments and form the adherent junction or the desmosome junction. So other types of receptors are also there, like Ig superfamily CAM, mucin like CAM. So CAM stands for cell adhesion molecule, selectins, integrins. So these are again a family of cell membrane-bound receptors that help in forming the cell-cell junctions. Now remember, if you, I think you remember, because I just told you, integrins are involved in forming cell and ECM junctions, correct? But integrins, some subset of integrins, they are also involved in forming cell-cell junctions as well. So like beta 1, alpha 4, beta 2, alpha. So these subsets of integrin receptors, they are involved in forming the cell junction types like you just saw. Okay. The cadherin family of protein on the selectin. This is again the family of protein that we just discussed, which are involved in cell-cell junction formation. They require calcium ion for binding. Okay, so which is why during cell culture, so we use a very important step called frictionization. Okay, where we separate out the cells from one another, means we break these cell junctions and create single cell suspension, okay, where each of the cells can be observed individually. So when doing that, we need to break these cell-cell junctions. Now, as I told you, the cadherin in the selectin family of protein that is involved in forming cell-cell junction, they require these divalent cations like calcium ions, which is why in the frictionization step, we use a reagent that has a chemical called EDTA. Look up its full name in the internet. So this is a chelating agent. It can easily bind to this kind of divalent cations and thus disrupt the cell-cell junctions. And then if we do that, we can then easily get single cell suspensions. So that is the role of EDTA when you are doing frictionization in cell culture. So we will actually see in one class, in very briefly, how to handle cells. Because when you are going to make the tissues, you need to isolate the cell from a patient or from an animal and then grow the cell number. And that you have to do it in a well controlled environment, a sterile environment. So how you do it, like what kind of media you need, what steps are important, how you store these cells. Okay, so we will take one very brief class. Actually, we'll cover up in one class only the some principles that you will need in future for doing cell culture protocols. So if you understand that during this course, in future you may be taught various kind of steps. You know, do this, do this, do this. So many people don't actually tell you the basis of those steps. They just tell you like they just want you to do रट्टा मार लो कि ये स्टेप करो फिर ये स्टेप करो ये स्टेप करो रट रट लो बस ठीक है most of the people do this. I have seen many people doing cell culture experiment by just remembering the steps that is involved. So, which is why they can't troubleshoot very nicely. So, if something goes wrong in any step, they don't know why and what to do to solve it. So, if you don't know the basics, why this step is needed, what is the role of this chemical. So, these things, if you don't know prior, then you won't be able to maintain your cells pretty nicely. And tissue culture तो बात की बात है ठीक है tissue culture engineering is like way different thing okay so that is why we will cover one class where I will explain to you some of the basic techniques that people do and what is the rationale behind doing it how do they do it I'll just explain okay later on so these are different kind of, as I said, membrane-bound receptors, the Ig superfamily CAMs, cell adhesion molecules. So here, different kind of domains are there that connect the cells. Also, mucin like protein, it looks like this. It has carbohydrate molecules. This basically protein that is conjugated with some polysaccharide molecules. And selectin molecules, these also require the calcium ions for binding with other receptor molecules and some kind of integrins. They are also involved in forming cell-cell junctions. Now, estimating intercellular flux. So you saw that in gap junction, the molecules are transporting right from one cell to the another. Okay. So what is the flux of this? Means what is the rate of movement of these molecules per unit area per unit time? Okay? Or what is the concentration of or at what concentration these molecules are moving okay per unit time and per unit area. That is the definition of flux in general. So the junction between the adjacent cells, they typically on the order of 1.5 nanometer. The diameter of this junction is in few nanometers. Okay? And allow molecules which are of molecular weight less than or equal to 1 kilodalton. Since these cell junctions they allow for direct exchange for molecules, it is of interest to estimate the flux that is achievable through them. Okay, and note that these molecules which are exchanged, they never leave the cells because it stays in the cell ultimately. It is just moving from one cell to another. It is not diffusing out, it is staying inside. And their size, what in the size is limited by this diameter, 1.5 nanometer diameter. Okay. So the maximum flux, again the definition of flux is the concentration per unit time per unit area. Okay, that is the definition of flux. So you can estimate the maximum flux by again uh, modeling it using by considering a model of flux through a hole on a flat wall. So if you have a flat wall and there is a small hole and some molecules are there on one side of the wall and on another side of the wall there is no molecule. So you are studying how fast or at what rate these molecules will pass through this hole. So this is again a mathematical model that people use. Okay, and the researchers who have estimated this maximum flux value, they have thought that this type of mathematical model fits best in this scenario, which is why they have used this model. So the equation that they get from this model is this. This equation flux is equals to 4d by pi d into C1 minus C2. And this equation they have just simply put and in this scenario, okay, to estimate the maximum flux of the molecules that is passing from one side to the another side. So the typical values, small d is the diameter diameter of the I guess molecule. Yeah, small d is the diameter of the molecule. Capital D is the diffusivity or diffusion coefficient. So it is in the order of 10 to the minus 5. So if you recall, in one of the classes we see that the diffusivity of very small molecules, like very small molecules, are diffuse at this kind of value. Okay. So for the small molecules, the diffusivity or the diffusion coefficient is taken this. Okay, and the concentration difference. So if the molecules are passing from one region to another region, where the passive diffusion is going on, right? So there should be a concentration gradient. So when this gradient is shown by C1 minus C2. So there is a wall. On one side there is C1 concentration of the molecule, another side there is C2 concentration. Difference is 100 micromolar. So if you feed these values in this equation, you will get a maximum flux of 2.4 × 10⁵ molecules per second per pore. Now here I said area, right? Now here the area is limited by the size of the pore or the number of the pore actually. So because there are so many pores that is there in between two cells. So rather than taking an overall area, they have just taken it as a number of pores, how many pores are there. So through each pore, this is the number of molecules that can pass through each second. Okay, this is the maximum flux that can be obtained. Now with approximately 100 pores between the cells, if you multiply this value with 100 pores, like you're assuming there are 100 pores between these cells. So the value will simply be increased by 2 orders. Okay. So you can now get between two cells, this is the maximum flux. Okay. Now here per pore is not given. This is the value for per pore. This is the value for the all total number of pores. So is there any confusion in understanding this expression? So this expression, how did it come? How did they derive it? It is none. It is not scope of this course. They have just simply used the mathematical model for understanding the flux through a hole on a wall, okay, and they've applied it into this scenario. So how did this equation come? You don't need to worry about, but you need to understand how the value of the maximum flux is obtained. What is the meaning of these symbols in this equation? Okay, why difference between the concentration is taken? What is the value of diffusivity? What should be the right value of diffusivity or diffusion coefficient that we should use? And here is the diameter of the molecule. So different diameter of molecule will change the flux, obviously. So if I ask you, the diameter of the molecule increases. If suppose the diameter of the molecule increases, what do you think? The flux will increase or decrease?
Ma'am, it will decrease.
Correct. It will decrease because it is inversely proportional. Similarly, if the molecule is having a very high diffusivity, means it has a diffusivity, remember that it's an internal property. Like, Young's modulus, so it is an internal property. It is defined for a particular type of material. Similarly, diffusivity is an internal property of a particular type of molecule. So proteins will have different diffusivity. ECM molecules, if you recall, they are very large to have, so they have very low diffusivity. So the diffusivity, if it increases, means the molecule has a less tendency to diffuse. Then the flux will increase. Okay. So you can think on these kind of interrelations. Okay. Similarly, if the concentration gradient increases, the value of concentration gradient means there is a very strong concentration gradient. One side there is lot of molecules, another side there is zero molecules. So direction of the movement you can obviously, I think you can obviously understand that it is from high concentration region to low concentration region. But in that case, the flux will be also high. The molecules will more number of molecules will pass through per pore per second. Now let's look at one example. of how cell-cell junction is helping, junction-based cell-cell interaction is helping in a process called leukocyte extravasation. So, what is leukocyte extravasation? When people observe it, why it is important? So, leukocyte is white blood cells, correct? Now, when there is an infection at some location in your body, then the leukocyte, the white blood cells, they are signaled to come and fight this infection. Now you know they are flowing in our blood. So and the blood is having endothelial lining. So endothelial cells are forming a continuous lining, okay, and forming the blood vessels. So these white blood cells which are flowing in the blood, they have to come and interact with these endothelial cells. And interaction will, and when a certain kind of interaction takes place, then only between two endothelial cells, the leukocyte or the white blood cells can pass through. And what kind of interaction takes place in this scenario? Their cell junction based interaction takes place. So this is a very interesting thing actually of how it is happening. Let me zoom in so that the figure is much more clearly visible. So here, this is a leukocyte and this is the endothelial cell. So endothelial cells have receptor bound, so membrane bound receptors like ICAM1. This is ICAM1 receptors. Then it will also secrete another soluble receptor, that is P-selectin. This is the soluble form of the selectin receptor that we have just seen. You know, a receptor can have a membrane bound form or a soluble form. So P-selectin is a soluble form. So when and nearby, this is a leukocyte which is having this integrin receptor, alpha 2 beta 2 integrin receptor. It is also having a receptor called PAF receptor, a platelet activating factor receptor, and there are some carbohydrate based ligands. Okay, so these receptors are there, membrane bound receptors are there in the leukocyte. Now they are going to help in this extravasation process because these receptors are going to interact with the receptors present on endothelial cells. How does it happen basically? So when a leukocyte reaches an endothelial cell, endothelial cell becomes activated. Okay, so once it's activated, it is going to secrete out the P-selectin, that is soluble form selectin receptor. And this P-selectin will go, this P-selectin molecule will go and bind with the carbohydrate ligands that are there on the leukocyte. This in turn is activating the leukocyte cell. And also it is trapping the leukocyte at this position. The activation of the endothelial cell is also causing secretion, the synthesis of the FAST molecule, platelet activating factor molecule. So once this molecule is synthesized, it gets expressed on the surface of the cell. So again, so this FAST molecule is going to attract the FAST receptor present on the leukocyte. So in this way, the leukocyte is trapped at that site through FAST receptor and the carbohydrate ligand that is present on the leukocyte. This is the leukocyte trapping cell. After it is trapped, it is activated. Leukocyte is activated. How? Due to the binding of FAST receptor and FAST molecule, these integrin molecules, alpha 2, beta 2 integrin molecules, they are also activated. Due to their activation, they can now go and bind with ICAM, one family of membrane bound receptor that is there on the endothelial cells. Once it is happened, the leukocyte is adhering with the uh, endothelial cells. So upon adhesion, now this cell can simply slide out from the endothelial lining. So basically, uh, a large pool of white blood cells, they have now escaped the blood vessel, basically oozed out from the membrane, from the endothelial wall, and now they can go either enter the lymphatic circulation. Okay, so lymphatic fluid also have a high WBC count, or it can just go and fight the nearby tissue infection. Okay, so if there's a tissue nearby the endothelial blood vessel, it can go and do its job. So this is the process of leukocyte extravasation, where the membrane-bound receptors are helping to interact between two totally different types of cell population, and thus allowing the leukocyte cell, to go and fight the infection at the tissue site. So let me again describe it using the points given here. The leukocyte extravasation through endothelial layer serves as an example to illustrate the bidirectional nature of cell-cell communication. Endothelial cell, it expresses ICAM1 receptor on its surface. Now, once it's activated, it's going to secrete the P-selectin molecule and as well as synthesize the platelet activating factor, which get displayed on its surface. The mobile leukocyte, they usually express alpha 2 beta 2 integrin receptor, FAF receptor, and integrin ligand, which is the carbohydrate molecule on its surface. So when a nearby mobile leukocyte, it comes near an activated endothelial cell, it slows down and gets trapped. Okay, so trapping will happen when the p-selectin molecule interacting with the ligand, selectin ligand, and FAF receptor and FAF molecule interaction has taken place. This interaction then triggers leukocyte activation that leads to activation of integrin molecules. Uh, Upon activation, these integrin molecules can go and bind with the ICAM1 family of protein that is there on the endothelial surface. So once interaction is taken place, the leukocytes can adhere on the endothelial layer and then extravasate into the interstitial tissue to fight the infection. So, is there any query in understanding these points, this process overall? It might sound like since you're studying for the first time, so it might be a bit confusing, but it's not actually. You can see few classes of proteins, they're interacting at different times, and because of which ultimately the leukocyte cell is able to move out of the blood vessel. So, any queries in understanding this process? Okay, I hope you have understood. If there are any queries, even after I have moved forward, you can stop me anytime and ask. Now, there can be some malfunctions in direct cell-cell contact signaling. So, if there is a defect in cell-cell interactions, like say correct type of protein that is not synthesized because of some mutation in his gene, so there will be a defective cell-cell interaction take place. So this can cause overall uh, malfunctioning in the tissue function. Some kind of skin diseases, like blistering skin diseases, they are caused by defects in the integrity of epidermal cell junctions. Okay. Then the antibodies against the cadherin family. This is a protein belonging to the cadherin family, and the defects in the cytoskeletal keratin network, they are both being linked to this class of skin diseases. So if there is some defect in the cytoskeletal keratin protein or the synthesis of the antibodies, uh, autoimmune disease basically, the body synthesizing antibodies against the own proteins, that is cadherin family. So if these things happen, then proper cell-cell junction will be not formed, and the disease like skin diseases, blistering skin diseases can take place. Other type of also there is defect in gap junction, means the molecules forming the junction. There is a defect in those molecules, protein molecules. So it has been seen in a disease called X-linked catgut Marie-Tooth disease. So here what happens is that the defect is present in gap junction of the Schwann cells. So this is one type of cell present in the nervous tissue. The Schwann cells they form the myelin sheath around the neuronal axon, and if there is this defect, it can cause demyelination, okay, and degeneration of the motor neurons. And overall, this class of disease is called X-linked karyotype disease. So here basically, you recall the structure of the neuron, correct? And there is like the cell body, and there is the axon. And the axon, it has a myelin sheath, and this sheath is made up of such one cells. Now, if there is a defect in the gap junctions between the such one cells, then this class of disease called X-linked karyotype disease can start like showing symptoms in the person. And cells can also respond to various kind of mechanical stimuli. So far we saw how cell-cell contact is affecting cell function, basically controlling cellular fluid processes. Then soluble signal is doing it, and as well as the ECM proteins can also do it. But people have seen that basically this is a very new area where people have seen is if the mechanical property of the substrate on which the cell is grown is changed, the behavior of the cell is also changed. So the new class of signal transduction that is called mechanochemical transduction takes place, where a different kind of pathway, cellular pathway people have discovered, which can translate any type of mechanical stimuli into biochemical pathways and cause changes in the cellular function. So this area is still in its infant stage, and many new things like people are still discovering. Like it is clear that the mechanical stimuli plays a very important role in the functioning of the cell. Like for example, for endothelial cells, the shear stress that the flow of the fluid is causing. So when the fluid flows, the cells in the endothelial layer they experience serious stress. So due to this serious stress, the pattern of gene expression in endothelial cells they change. So people have observed it, as well as the compressive forces. They also alter the matrix composition that we have already discussed previously. That chondrocytes they can change the ECM composition when compressive normal forces are applied, and uh, depending on the direction of the fluid flow, how the fluid is flowing, then that can direct how the cells are going to cell in suspension. They are going to adhere on the flat surfaces. So this kind of fluid related research where flow of the fluid is directing cell activity becomes very significant when you are culturing large amount of cells in bioreactors. Okay. So in that case, if you want to know more deeply what is happening at cellular level, then you can go into this field of mechanotransduction, where people are studying or still and trying to understand what are biochemical pathways are responsible for translating the mechanical stimuli to chemical signals. Okay. And between the signaling pathways that we have seen, direct cell-cell, then soluble signals and ECM signals, there can be crosstalk between the different signaling modes. Okay, they can affect one another. They can affect uh, the functioning of one another. So we will see few examples of what is happening. Like how this crosstalk is affecting cellular functions in few cell types. So first, we'll see the crosstalk happens between soluble signaling and ECM signaling. So the endothelial cells, when they progress through the cell cycle, they do it under the influence of saturating amounts of growth factor. So cytokines are needed for cell growth and differentiation. So when the amount of the cytokines are at a very high level, then only the endothelial cells will be triggered undergo cell division. But this will only happen when the cells are spread on a laminin coated surface. Okay, so if the surface is not coated with laminin, the cells will not respond to the saturating level of cytokines and the cell cycle will not happen. Similarly, the cells that are not attached to an ECM cover surface but exposed to growth factor, they will undergo apoptosis rather than cell growth. So if the ECM component that I'm talking about, the laminin, which is an ECM component, that component is not there, but you are just giving growth factor, then rather than going through the cell cycle pathway, the cell will begin the apoptosis. So this is how the soluble signaling, that is signaling to the growth factor, is being influenced by the ECM component and vice versa. Then there can be crosstalk between cell-cell contact and the ECM signaling. So in case of hepatocyte, this example is for hepatocyte. Previous example was for endothelial cell. So the hepatocytes, when they are suspended in a collagen gel. Okay, so the ECM component here is the collagen. They function better in aggregates than as single cells. So aggregates means many cells are aggregated together. They are forming spheroid kind of a structures. So when spheroids, let's say, are suspended in collagen, they function better rather than suspending single cells. That is the how the cell-cell contact is influencing the functioning of the cell in presence of an ECM component. Similarly, if the collagen density is very high, okay, around the aggregates, then it also prolongs the stability. Means this hepatocyte function will be very much stable for a longer period of time. Then this shows how the collagen is in turn positively influencing the functioning of the cell aggregates. So if there is something else other than collagen if you use, okay, then you may not see this kind of a effect because here collagen is playing an important role or is somehow influencing the cellular contacts or signaling to cellular contacts. Okay? And vice versa. Cellular contacts is better cellular contact is happening in presence of collagen. Where in the third case, where there can be a crosstalk between cell-cell contact of cellular and soluble signals. Okay? Cell-cell contact and soluble signal. So this example is not for a particular type of cell, but rather it is saying that the gap junction expression means the proteins responsible for forming the gap junction, their expression is more sensitive to the hormone concentration. So hormones are involved in endocrine signaling. So if a particular type of hormone is present, then only a particular type of gap junction protein will get expressed. So they are somehow interrelated. So people have observed it. So these are some examples to show you how the different pathways of signaling, cell signaling, can influence one another and thus influence the cellular function. So this kind of knowledge is very much important when you are dealing with this type of cells and then bringing these cells into a artificial tissue structure and then overall trying to understand what factors influence the function of that tissue. Okay, so that's it. for the today's class. And in today's class, we have also finished a very big chapter actually. And from next class onwards, we will, as I told you, we will discuss about the cell culture techniques, the principles behind those steps that we do. Also, we will see what are characterizations that you need for to observe, like say microscopy, then different type of assays, etc. Okay, so these characterizations are also important because that helps you understand whether you are going right or wrong in forming your tissue of interest. Then also we will look into stem cells. Okay, what how the stem cells functions and different types of stem cells, etc. Because it is being very widely used in tissue engineering, especially for bone healing applications, etc. And we will also see how you will make your artificial ECM, also called scaffold, through various kind of biofabrication techniques and using different kind of materials. So we will cover that. And lastly, we will see the large scale production of the cells using bioreactors, how to get, say, millions of cells that we talked about, right? Or even billions of cells, which is the clinically meaningful number of cells that is going to be essential for creating a clinical benefit of your product. So you need that many number of cells in a shorter period of time, and you can only do it by using bioreactors. So how to do that? Okay. So what is the principle behind these kind of bioreactors? So with this, we will finish up this course like within 2 months, like September and October. So already 2 months is passed, 50% syllabus is done, and rest 50% is left. So any queries you guys have, now is the right time to share them. Were you able to understand today's content, or is there any issue in understanding any particular topic? Am I audible? Hello, is anyone there in the meeting?
Yes, ma'am.
All right. So is there any query in understanding today's content?
No, ma'am.
Okay. So I was just marking your attendance. So let me confirm if I have marked for everyone who are present. I have marked for Molly, Keetan, Janvi, Vani, Harshita, Aditya, and Nidhi Priya. Is anyone else left? Seven of you. So yeah, seven people is been marked. So is there any query related to the quiz question? So how was the quiz? Like, how would you rate the questions? Were they easy, difficult, attemptable? Like, uh, how was the quiz question paper?
Ma'am, moderate.
Huh? Moderate. So, after seeing the solution, have you guys seen the solutions? solution that I've shared over mail? And are you guys satisfied with your marks that you have obtained? Or you think you could have done better? So Vaishnavi, are you facing any internet issue? Because I'm seeing that you're
Yes, ma'am.
leaving.
Yes, ma'am.
Okay. So I'm also marking your attendance as well. I think Shambhavi
Ma'am,
is has not
Shambhavi is
joined.
also phasing.
Okay. So
Ma'am, she texted me.
okay. So was she present during? Actually, I didn't see. Was she present during the sessions? Like I didn't see a
No,
live.
ma'am. No, ma'am. She was facing problem from the starting of the class.
Okay. Okay. So ask her to see the video, recorded video, so that she will be able to
Yes, ma'am.
understand it.
Yes, ma'am.
So I'm marking her attendance as well. So I was just discussing, like, how was the quiz? Are you still satisfied with your marks, or do you think it could have been better? Or if you have any query related to any question, or the math problem that I gave, if you have difficulty in understanding the logic behind those solutions. So I hope you guys have gone through the solution, right? Because that is more important than the marks. Understanding is more important. Learning is more important. Whether you have learned something, that is very much important. So, do you guys want me to pick someone up, or voluntarily you will reply?
For what, ma'am?
I was asking, like, how was the quiz performance?
The
What do you think
quiz
was it?
was okay.
Okay. So, if, do you have any doubt in a particular question?
Sorry, ma'am.
So, you have gone to the solution, right? Solution was understandable.
Yes, ma'am.
Okay. So, I hope this is the response from everyone else. Or does anyone have any different response? So, I'm assuming that is same for the others as well. Try to so I would say the hard part of this course is over, I would say. Okay, till now, till today's class, the content that you were seeing, it was a bit challenging, I would say, and very new. From next class onward, you will see it is much more relatable. Okay, means you might have read it somewhere previously. So when you will go through that content, it will, and you will be able to understand better. Okay. And are you guys still working on the project? Have you guys started working on the project, or still waiting for something? Are you having?
Group
Yeah,
B
We are
started.
working.
you're working. So are you? Have you made WhatsApp groups? How are you discussing, like weekly meetings or anything like that? What's going on?
Yes, ma'am.
Okay, so you are doing meetings.
Ma'am, we have created WhatsApp group and through that we are contacting each other.
Okay. So, whatever content that each one of you is reading, you can start writing, like summarizing those papers yourself, so that in the end, when individually you guys will have the content, you can just sum it up in a short report. Okay. It will also help you to, in future, if any one of you goes for research, like any research position or a PhD degree, if any one of you goes then this kind of exercises like understanding literature, summarizing literature, this will really help. So is there anything else you guys want to ask me?
Ma'am, next class you can give a brief about how you end up PhD and how you got this PMRF scholarship like that. You can say it like that.
Okay, so yeah, definitely. I'll give you brief like about my journey so far, like my degrees and where did I do it, and PhD degree, how it's going. I'll give you. It will be. I should have done it initially, but yeah, I'll do. Thanks for bringing
Okay,
it up.
ma'am. No problem.
So we'll meet tomorrow around the same time, 7 p.m. And tomorrow we will discuss the Article 3 that I've shared. I should have discussed it last week, but due to some work I couldn't. So this week we will discuss it. It is on wound healing basically, and it's a recent paper. Try to go through that paper once. Don't worry if you don't understand. We will ultimately discuss it. And if you have any queries, you can bring it to tomorrow's class. All right. Thank you for joining today. You guys can leave the meeting.
Bye, ma'am.
Bye.